Now, a platform trial can combine elements of a basket trial or an umbrella trial. Its major advantage is that built into the initial trial protocol, which is the platform trial protocol, is the ability to adapt. And that adaptation may be, for any given drug that passes a threshold of activity, to suddenly advance to a larger trial or even a randomized trial. But more commonly, it's adapted to allow sub-studies with new drugs to be added over time as those drugs become available, assuming they are relevant for the patients the trial has been designed for. Now, these designs have been utilized to evaluate different types of targeted drugs. Those targeted drugs have largely been ones that are targeting a particular gene variant or the protein product of a gene variant or pathway. They include the NTRK fusion genetic variation, because there is a small group of cancers that have that genetic variant. They include pembrolizumab in patients whose tumors have mismatch repair deficiency, which not only confers an inability to repair DNA damage, but confers a sensitivity to an immune checkpoint inhibitor. And pan-BRAF inhibitor, RET inhibitor, these are some of them, but not all of them, that have been commercialized. The things to consider, first of all, the only one that was truly developed from the very beginning as a histology-agnostic trial that led to drug approval based on a large phase 2 single-arm experience was the first one, the NTRK targeting drug. And the reason for that is because NTRK is expressed in 1% or less of all cancers. But when it's present, it is the key driver mutation. It can be targeted, and there is a very high response rate, about 70 or 80%, and those responses are durable. And when compared to historical data from patients who have been identified to have NTRK fusions from retrospective evaluations of tissue and clinical outcomes, this is clearly better than anything had been seen before. All the other drugs first were developed and got their first indication following a traditional cancer drug development pathway, and subsequently were evaluated in these histology-agnostic pathways. And I think that is probably because there was a histology-specific pathway that was feasible, there was an unmet need, feasible to execute, and there was, if you like, the science behind developing it in a histology-specific way. So there is a balance, if you like, between those particular factors, interesting, relevant science, feasibility of execution around a traditional or less traditional, but where there's precedent in a histology versus agnostic pathway, and the clinical need. Those are the key determinants that go into how you might design and execute a trial. Now, for histology-agnostic trials, you need to have an understanding of the prevalence of the expression of the common biomarker. The neat thing about theranostics, which you all know, and I certainly appreciate, is it doesn't matter if that is actually contributing to the propagation or the survival of the cancer, as it does with the approaches used with small molecule therapies. It's really about, can it be exploited as a delivery mechanism for the radiotherapy? And that's a key advantage, I would say, for tumors that are predicted to be still radiation sensitive or likely to be radiation sensitive, even if they have become refractory to drug treatments. So the expression of the target, whatever that target is, must be identifiable and exploitable with your radiopharmaceutical, and you're leveraging that targeting and cross-histology delivery and hopefully sensitivity of your treatment. So if you like, it is really, as I think was said earlier, a guided missile approach. It has all of the markings of a precision medicine strategy and a potentially very successful one. Because those protein targets that can be identified, have been identified for radiopharmaceuticals, which I've tried to summarize some of them here, are expressed across different tumor types to varying degrees, and that expression is greater than in normal tissue, and the delivery of agents that can engage with and subsequently the cells that can be irradiated from the radiopharmaceuticals suggests that there could be broad benefits that could be identified in a histology-agnostic trial that would be more efficient than trying to mount individual trials in each setting. So the scientific rationale and the clinical need are there, that key first principle, if you like, in terms of should we go ahead with a trial and how should we do it. There are relevant biomarkers. There is the ability to develop the imaging tests to screen for the presence of those biomarkers as a way of identifying your patients. There is a need to understand the prevalence of that expression because there is an efficiency and cost with screening that has to be considered carefully. There's assumption that the radiation effect would be independent of histology. And in some cases, that may be quite relevant. In other cancers known to be radio insensitive, maybe not. So you can't expect the same response rate in all settings, either because of the inherent radiosensitivity of the target may be different, or it may have changed over time, as does the expression of the target itself. Radiosensitivity of the cancer cell, of course. And certainly there's the opportunity to exploit rare cancers, where individuals with a rare cancer generally don't have clinical trial options because it's prohibitively challenging to mount a trial in rare cancer settings, but they could be eligible for these trials. The next step is to pick your design, generally the most straightforward design, the one with precedent, the one with the least complexity is a basket design. And this is where the fact that patients would generally be those patients whose disease had progressed through standard therapy would be the eligible patient population regardless of the cancer histology. They have no alternative options at that point known to provide benefit, and so you don't have to do a randomized trial in this setting, but you do have to have some understanding of historical control data of outcomes for these patients in these settings. Other trials could be considered, but they have additional complexities, and generally speaking, would have to be filling a particular need to be considered. Now, there are no precedent radiopharmaceuticals that have been approved for a histology-agnostic setting, at least that I could find. But there are those that have been discussed, particularly among academic investigator-led cooperative groups like mine and others, and there are those that are moving forward, beginning with histology-specific results, but with planned expansion into histology-agnostic areas. And I've just summarized them here. So I am sure there will be histology-agnostic radiopharmaceutical trials, and I'm sure that there will be an indication and approval emerging from some of these trials. What you need, and other people have touched on this in the last session better, but just to highlight, you need an infrastructure for the testing, you need an infrastructure for the delivery, and you need an infrastructure to be able to coordinate all of that across the different sites that are participating. You also need to have a good discussion with your regulators, not just in Canada, but in other jurisdictions where the trial may be running, but also where one hopes to get the commercialization of the product. And different regulators have different criteria for their clinical trials and the requirements for the conduct of those clinical trials that you need to be aware of and adjust to, as well as what they would be looking for the commercialization package of evidence. So if we think about challenges and considerations, you want to pick a target that it's expressed relatively similarly across different types of cancer. You want it at a level that your screening for the presence of that target is efficient and not too costly. You want to be able to do the dosimetry, define your regulatory pathway and your reimbursement pathway, since those are separate in this country, unlike some other jurisdictions. You need to develop the radiopharmaceutical as a drug and the companion diagnostic as a diagnostic. Both of those criteria have to be satisfied from the regulatory perspective, and the whole aspects of manufacturing and delivering the radioisotope, all of that operational complexity needs to be addressed. And your centers have to recruit patients and be able to deliver and manage the radiopharmaceuticals. Now, I won't go into details about this because I'm running out of time, but and I should never be talking as if I'm a regulator, because I'm not. But there are these differences in terms of what the FDA looks for. They have a guidance document, and Health Canada looks for, which is generally they're following radiation safety protocols and their drug regulation criteria. Operationally, there are essentially checklists that one can follow to credential your sites, as well as to make sure that your trial, as sponsor doing the oversight, is conducted in the appropriate manner with all of the aspects required for the execution. The statistical design for a basket trial, the one thing that I would highlight is you do need to account for a certain number of patients with each of the defined histologies to the extent possible. So a basket trial essentially has a series of subgroups based on histology that are large enough that the level of activity, usually tumor shrinkage being the endpoint and the duration of that shrinkage, can be assessed. And if that is showing some consistency across histologies, you don't have to do it for all histologies, but you have to convince the regulators it's likely true for other histologies as well. The strategic opportunity to move forward in Canada with the development of radiopharmaceuticals for cancer in particular, I think is real and quite compelling. We have the leadership across all of the elements to be able to manufacture, develop, conduct, and deliver for regulatory and reimbursement review the evidence for the benefit and safety of the radiopharmaceutical. There are multiple trial development opportunities and example trials of histology-agnostic cancer trials of targeted agents, precision medicine, that have led to approval. So we have a pathway here. And we have multiple collaborations, which we've heard previously are really key to the execution of those trials. So we have the leadership, we have the skill set, we have the collaborations that can move forward. And so with that, I expect we will see histology-agnostic radiopharmaceutical trials. They will capitalize on their precision medicine properties that will facilitate and enable their success. They do require work and changes, because the biggest threat, I think, to clinical trials of radiopharmaceuticals is the inertia in the system that maintains the current status quo, where we need new collaborations between, for example, nuclear medicine and the oncology community, and probably pathology as well, to make them work. But they have the high potential to improve outcomes for patients. For many patients with different refractory cancers, and including rare and difficult to treat cancers. So thank you very much. Rebecca, what do I do next? Oh.
So maybe this isn't a fair question because it's not really about clinical trials, but I get asked all the time about all of the patient data that we saw come out of Germany, where these are not part of clinical trials, but they were really compelling data points that really increased the interest in developing new radiopharmaceuticals around PSMA. Do you see a role for that kind of program in Canada? Like, how could it fit into how Canada advances innovation within kind of, of course we want people to be safe, of course we want all of these things to be regulated appropriately, but is there a place for that kind of innovation in Canada?
I definitely see the advantages, and I think people here would probably be even more familiar than the German regulatory and legislative facilitation of what are essentially low-dose radiotherapy studies. Also, they have enabling legislation for microdose studies of many different types. They're very big on homeopathy as well. I don't know if that's related. In the US, as you probably know, there is legislation and regulation that enables microdose studies as well. I think that even though it requires a big step, it would have to be a change in the regulations and the interpretation of those regulations to enable them. It's pretty clear that they do facilitate at least the initial, presumably low dose and safe evaluation in humans. And after, again, 25 years in cancer drug development, the only experimental model that I really believe in is the human one. So I do think there is a potential role. It's a potential challenge as well.